This video covers the IGCSE Biology Objective 6.2.2 on the 2025 CAIE syllabus to identify structures in a dicotyledonous leaf from diagrams and images. So here we have three different ways of looking at leaf tissue. We have a diagram on the left, we have an electron micrograph in the middle, and we have a photomicrograph on the right. So these are three different images that you will need to be able to look at, understand that it represents the microscopic layers of leaf tissue, and then be able to identify the various tissue within that specific type of image. Let's just take a look at the structures that you will need to know. So we're going to use the diagram, and then we'll look at the same structures in the micrographs. So first of all, on the outside, on the upper outside surface of a leaf, we have what's called a cuticle. This is not actually part of a leaf as in physical tissue, but it is part of the upper component of the leaf, so you will need to identify that. The first layer of cells is called the upper epidermis. You will see that they do not contain chloroplast. The second layer of cells is called a palisade mesophyll cell. You should recognize this from when we talked about cells in the cell organization unit, chock full of chloroplasts. Then we come to the spongy mesophyll cells, not quite as much chloroplast and having a slightly different shape. Then we have the vascular bundle, which is going to contain xylem and phloem, and we'll talk about that a little bit later. We have air space between the spongy mesophyll cells. We have the lower epidermis of the leaf. This is the bottom part that is not facing the sun. Again, not containing chloroplast. We have guard cells, which do contain chloroplast. They are located in the lower epidermis, and we'll take a look at what that looks like a little bit later. And then we have the gap between a pair of guard cells. This is called a stoma, singular, and stomata, plural. So let's take a look at these same structures now in the electron micrograph and the photomicrograph of microscopic images. So again, we have the upper epidermis at the top that faces the sun of any leaf. We have the palisade mesophyll cells directly below that. We have the spongy mesophyll cells directly below those, and the vascular bundles, which would be spread out. There would be many of these within that layer of tissue. We have the air spaces, and we have the lower epidermis with guard cells and stomata, which I have not labeled here. All right, let's just take a look at the lower epidermis. So we're looking at a photomicrograph. If we took a leaf and flipped it over upside down, we'd be looking at the bottom of the leaf, and this is what we would see. So this is the lower epidermal layer, or the lower epidermis of the leaf, and we can see three different components from the previous slides that we looked at. Number one, we have the epidermal cells that are just basic, transparent, no chloroplast inside of them. We have pairs of guard cells, and you can see they're all paired up as they are spread throughout the lower epidermis. They do contain chloroplast, so they are going to do photosynthesis, and we will explain why a little bit later. And then finally, we have the stomata, singular stoma, and those are located within the pairs of guard cells. And this is what it would look like if you were to look at the broad underside of a leaf. Finally, let's take a look at the vascular bundles in a little bit more detail. So I have some images here, drawings and photomicrographs. So the first thing that we need to understand is that the vascular bundle is either the midrib or the vein. And you can see in the diagram here that we're looking at two different sections that have been kind of colored in, the red and the blue. So there's a section that is towards the upper side of the leaf and a section that is towards the lower side of the leaf. So sort of facing the sun and away from the sun. So the upper side of the vein or the midrib will always be xylem. The lower side of the midrib or any vein will always be phloem. It always will be that way. And when we talk about transport in plants a little bit later, we will learn more about xylem and phloem. But for now, we just need to know where they're located in leaves. So the xylem is going to be towards the upper epidermis of a leaf, as depicted here in both of these photomicrographs, and the phloem, looking slightly different, will be located towards the lower epidermis of the leaf. And this is how they're situated, whether it's the midrib or any other smaller vein in the leaf.